All right. So, hello everyone. I'm Aushandra, and today we are going to discuss. We are in the fourth lecture now, and we are going to discuss a paper basically that talks about the application of extrusion-based 3D bioprinting. All right. I have already shared this paper with you on the Google Classroom platform. You guys can later go ahead and download it. You can see it under classwork and it is available as a reading assignment. So in your free time, go ahead and read that paper. It's a very short paper and it's very easy to understand after uh, you have done with this lecture. Okay, it will be easy to understand for you. And I'd also publish an assignment so that based on this paper only, and it will definitely help you then. So quickly, since we have already seen what is extrusion-based 3D bioprinting, what are the modes, but quickly recap about the technique. So, in extrusion-based 3D bioprinting, we make something called bio ink, right? Which has a type of rheological property. That property is called shear thinning, right? We studied that. Just quick recap first. So it should be shear thinning in nature. What is shear thinning? Means its viscosity decreases when shear forces are applied on it, and when you remove the shear forces, viscosity increases again. So that is the shear thinning property of this bioink. These are loaded into printheads, and into this printheads there is an extrusion system. All right. So this extrusion system can be like based on air, that is pneumatic pressure is applied for on the top of the bioink, or mechanical forces are applied like a piston or a screw in order to extrude it out from the nozzle. So this nozzle is typically of a very small diameter, and because of which you get very thin filaments coming out from the tip of the nozzle. All right. So that is extrusion. And when you assemble these filaments layer by layer, you get a 3D structure, right? That's the basic understanding of how the technique works. And when you add some biological component like cells or growth factors or spheroids, anything that is biologically derived, even the FBS, fetal bovine serum or anything, you make something called bio ink because it can now support the growth and culture of cells, and you can transplant it on your body because it is safe now. Okay, now coming to the application. So for understanding how this technique is applied in research, I have taken this paper for our discussion. So this paper was published in 2017 in a very reputed journal, Advanced Healthcare Materials. All right. So this is one of the first papers that I read when I started my work in this area. That is why I wanted to take it up. And I found during that time I understood this paper very well. So since you guys are all beginners, so that's why I chose this paper. Okay. So go ahead after this lecture, read this paper. I have already uploaded it in the assignment section, classwork section, and you can download it, okay, and read it afterwards, okay? But do that. It's a very nice paper. So this is the title of the paper, Human Skin 3D Bioprinting Using Scaffold-Free Approach. Okay, we will understand why they have used the term scaffold-free in a minute, and what they have made. They have made a human skin using this extrusion-based 3D bioprinting technique. Okay, so if you have any queries in between, feel free to ask. So that's already understood, but I thought to remind. Okay, now let me quickly read the abstract of the paper. Okay, so that you get a general overview what the paper is talking about. Organ in vitro synthesis is one of the, maybe there is a laser pointer of some sort. Oh no, there is not. Okay, just just follow along. All right, just follow along. Organ in vitro synthesis is one of the last bottlenecks between tissue engineering and transplantation of synthetic organs. Bioprinting has proven its capacity to produce 3D objects composed of living cells but highly organized tissues such as full thickness skin. That is epidermis and dermis. They are rarely attained. The focus of the present paper or present study is to demonstrate the capability of a newly developed ink formulation and the use of an open source printer for the production of a really complete skin model. The proofs are given through immunostaining and electrostatic microscopy that the bioprinted skin presents all the characteristics of human skin, both at the molecular and macromolecular level. Finally, the printability of large skin objects is demonstrated with printing of an adult size ear. So, quickly, what the abstract is saying or what the paper is talking about. Paper is saying that at the time that it was published in 2017, this technique, extrusion-based 3D bioprinting, it was very recent. Like it was the newest topic at that time. So at that time, people have started thinking of the potential of using this technique for making complex tissues, but they haven't really made a complex tissue like skin. Okay, that too with both the layers of skin, dermis and epidermis. First thing, first novelty that it presents. Second, they're saying that they have developed their own bio ink and they made their own 3D bioprinter. Okay, and using these two systems, they made a complete skin model. Then they showed the completeness of the skin model, means how much the skin model mimics the real thing, the native skin, through immunostaining, okay, or it is also called immunohistochemistry, like they do using antibodies and all, and they stain the specific surface markers or specific proteins that are characteristics of particular layers of the skin. We will see that result. And as well as they have done transmission electron microscopy, TEM it is called, to show the features of the epidermal layer. All right, the very intricate features of the epidermal layer. Okay, and to prove that the epidermis has perfectly formed. And lastly, to demonstrate the capability of their developed bioink, they have also printed a very large sized ear. Okay, so that is the overview of the study or um, the gist of the paper. Any questions so far? Okay, moving on. So let me start the introduction of the paper. So why did they need, why did they feel the need to bioprint a skin? Why? Like, we could have like taken, a small section of skin from a human and used it. Why to bioprint it? So that's the question. So first, so there are a few advantages to it. Okay. First advantage is that you can use those skin model to test the safety and efficacy of substances or drugs. Substances, I mean the drugs that pharmaceutical companies make. Okay. And then use it for fundamental research as well. So it can accelerate the development of various cosmetics and creams and other drugs to come to market faster, and also reduce the cost in making those drugs as well. So it can basically facilitate the research, entire differently research area, entire different research area, that is drug development and drug screening and all. even cosmetics development. Another thing to note is now many governments all over the world, even in India, they have banned the usage of animals for testing cosmetics. So if is I don't know if you guys buy cosmetics regularly or not, but many of those brands they say that they are now cruelty free, means they are cream or gels or whatever shampoo etc. They are not tested on animals now. Previously, they used to use rabbits and large animals like pigs and all, and it was considered cruelty, right? You test different chemical formulations on them. So now all those things are banned, and even for drugs that comes to India now, the Indian government has made a rule that you don't have to test the drugs on Indian people again if it is already passed a clinical trial in a different country. Okay, so if so you don't have to do the entire research in India again for introducing that drug. So, and also you, if that drug has passed all the things on animals and humans in different country, it is Indian government kind of like, okay, fine, go ahead and use it. So in this way, they are also trying to minimize the preclinical and the clinical research on those drugs altogether. So yeah, means minimize. So ultimate aim is to minimize animal cruelty. So there are various bodies like PETA who are actively working on that. So if animals cannot be used, so there are also various disadvantages using animals, like their physiology and anatomy is not completely same as the human. So those drugs or cosmetics that pass through preclinical trials or show good results on mice and rats, they often fail on humans, right? So there is actually advantage now if you stop using animals for research. But what's the alternative to that? Then comes like these kind of 3D tissue models, which are made with human cells. Only thing that we take from human is cells. And sometimes we also extract biomaterials like decellularized ECM, and that too from discarded tissues like placental tissue and all, okay, or discarded tissues that come out of surgeries. So you can use those tissues to make your biomaterial substrate into which you can disperse the cells. So these are what do you say, advantage to using tissue models. So first advantage is that you can use it for assessing safety and efficacy of substances. Second, that they can act as an alternative for animal models and basically accelerate the research on drug and cosmetics development and screening, right? Now, another reason why there is a need to bioprint for say, so people have been making 3D skin models before 3D bioprinting came along, all right? So people have made skin models using synthetic substances like nylon, right? So they just electrospun nylon or any other fabric-like polymer, and onto which they seed cells onto it. Okay, so it's like a textile type of thing. But the disadvantage is that substrate is itself synthetic. So it is not very biocompatible, right? It can show later, what do you say, side effects on implantation, or it does not fully mimic what is there in the body. Our substrate, we do not have any traces of nylon or anything like that in your tissue, right? So there are organic materials like collagen and all present. Another type of skin models that they have made is just using protein, okay, no cells. So only a layer of collagen, and they are saying that it mimics skin. So those kind of models have been also been made. But there are some drawbacks to it. What? These models that they make, it takes a lot of time to make it. Okay, and also the geometry in which they are made, they are very like predetermined. You can make only certain types of geometry, like planar geometries, flat geometries, that's it. But when you talk about wound sizes or the geometry of the wounds and all, it does not have to be flat all the time, right? It can be, it can have a very irregular shape. So these kind of first generation skin models cannot be very well applied to those wound sites, as well as this, since they use multiple steps, like first they have to make the layer of the nylon per se, and then seed the cells. In bioprinting, you mix the cells and the ink together in one step and do it, right? So so less time to make a skin model, and also you can make very unique geometries by bioprinting. So that is why we need to bioprint. So any questions till now? All right, no questions. Moving on. So let's start with the essentials, 3D bioprinting essentials that they need, need for the research, for the work that they have done. So they have made their own extrusion-based 3D bioprinter system. So it's an open source machine. They have made this in their lab, all right? And it's probably much cheaper than the commercial ones. I think we already discussed this in our first lecture, right? That you can make your own 3D bioprinter, and the codes and all the softwares, etc., that you would need, they are like they are available in pieces individually over all over the internet. You just have to find them and stitch them together so that you can make an entire system. Okay. So this is the image of the bioprinter uh, that they have given in the supplementary section of the paper. Okay. The next essentials that they need for the 3D bioprinting is that bio ink. So the bio ink that they made is actually a three component ink. We already talked about it. You can make a multi-material bio ink systems, right? When we talked about the introduction of the extrusion-based system. So three materials that they used are gelatin, alginate, and fibrinogen. So we already talked about previously the how the alginate works. You remember? So alginate is a polysaccharide or a carbohydrate-based polymer that can form hydrogel in presence of some divalent cations like calcium, barium, etc. And gelatin is a protein-based polymer. It is derived from collagen. Basically, is collagen is partially hydrolyzed to get gelatin, and the material, it shows a phase transition. Means at 35 degrees Celsius, sorry, at below 35 degrees Celsius, the temperature is 35 degrees Celsius, less than that, then it forms gel, hydrogel. If the temperature is greater than 35 degrees Celsius, it stays in a solution form. So it shows a phase transition. Okay. But it has a very good shear thinning property. So it's a good shear thinning property. Means it if you use a gelatin only for extrusion, it will extrude very nicely into good good forming continuous uniform filaments. Okay. So whereas fibrinogen is actually again a protein, ECM protein, naturally found in our body. So you see when we get cuts in our skin, and you see a black colored clot forming after a few like few hours and all, it is because of this protein fibrinogen. So there is an entire clotting cascade that activates some enzymes, okay, that those enzymes help fibrinogen solution, like they are present in kind of a non-gel form, all right, or solution form, I think, in the body. And then in presence of those enzymes, this fibrinogen converts into a gel form. Okay. So combining these three unique properties of three different materials, they have made a bioink. You will realize how these materials are actually supporting each other's, like combating each other's drawbacks and strengthening and showing the strength of the property, individual properties. How? I'll explain to you. So gelatin, why they have used? They have used as a rheological component because I already told you it has a very good shear thinning nature. And the component gelatin they have added just to give strength to the ink after the printing is over. So they are printing on a cooled substrate onto which the printed filaments will deposit, and after the printing is done, the gelatin is still in soluble state. It is not chemically cross-linked. It is only physically cross-linked in presence of temperature. Okay, so if you increase the temperature, it will become soluble. So after it is printed, after the entire structure is printed, gelatin is solubilized and eliminated. Okay, then alginate they have added as a structural component because if you use a calcium chloride containing bath or something, then alginate will form a hydrogel. So it will give structural rigidity to the entire structure. So after the gelatin is removed in subsequent steps, alginate is what remains. But note, alginate does not have any property itself to allow cell adhesion. Since this is a plant derived polymer and plants don't have, the same kind of proteins and sites like the proteins available in mammals. So alginate cannot support the growth of mammalian cells. So for to combat this drawback, they added fibrinogen. So fibrinogen is again acting as both structural and maturation component. So fibrinogen is a glycoprotein. Please note, means it has the property of both a carbohydrate and a protein. So it has the entire backbone is a protein backbone, and it has some carbohydrate moieties attached to it. So it's called glycoprotein. So this fibrinogen can form a hydrogel that is called fibrin. So fibrinogen, it can form a gel that is called fibrin in presence of the enzyme thrombin. So when you get a cut on your skin, then a clotting cascade starts. That is the first step of wound healing. Okay. So in the clotting cascade, this enzyme is activated. Otherwise, it's in a non-activated form, okay, an inactive form. So when this enzyme is activated, thrombin goes and reacts with fibrinogen to form a fibrin gel. So this fibrin gel forms a transparent layer over the compromised barrier, right? So this fibrin gel they are using as a structural component. And why maturation component? Because it has sites. It has specific sites that mammalian cells can recognize and adhere to and then grow. So that is why it is called as the maturation component. You see, gelatin can also act as a maturation component, but it is not being chemically cross-linked. So if you uh, culture this entire ink at a higher temperature, that is at 37 degrees Celsius, you'll see gelatin will start coming out. It will not remain inside the ink. So that is why they had to use another maturation component like fibrinogen in order to support cell growth and proliferation and all in the later stage of maturation. Okay. Any doubts in understanding why these three components? Yeah, go ahead.
Can you give a exact idea about what is exactly hydrogel? So hydrogel is a single substance or it's like a property. Any substance can be a hydrogel.
So What is hydrogel? That is your question, right?
Yeah.
Okay. So hydrogel, the base of a hydrogel is a polymer. First understand that. And you know what is polymer? It is like you have monomer molecules, they are stitched together by covalent bonds and you get a polymer. This reaction is called polymerization reaction. So the base of a hydrogel is a polymer. Now, can all polymer be a hydrogel? Answer is no. Which kind of polymers will you term it as hydrogels? So the polymers which can retain large amount of water. Only those polymers are given a special name, that is hydrogel. Okay, so if you try to look at the structure of the hydrogel in SEM, suppose, you will see there are polymer chains like this, and they are covalently cross-linked to with each other. And these polymer chains have some hydrophilic moieties like say oxygen, amino groups and all that can like form very nice hydrogen bonds with water molecule, because of which now water can stay in between the spaces of the like the network, polymer network. Got it? That is hydrogen, nothing else. So you can term a polymer as a hydrogel only when it can retain a large amount of water. So if you put a block of say two type of polymer, so let's say PCl and gelatin, and you want to check which of these polymer is a hydrogel, you make small blocks out of it and put it in a water bath and leave it for, let's say, few hours or few days. What you will see after few days, if the size of the block is same, means it has not absorbed any water and retained it. If the block has increased in size, you can measure the size using scale or something like that. If it has increased in size, that means it has absorbed a lot of water, and that is hydrogel. So usually what we observe is that gelatin will swell. That property is called swelling. Gelatin will absorb almost 2x its weight of water and increase in size. Okay, whereas PCl, what we observe is not a hydrogel. Its size will stay as it is. Got it?
Okay, ma'am. Got it.
Yeah. And now, do you understand why hydrogels are used with cells? Do you understand the logic behind using hydrogels in the bioink now?
Because the any growth factors or other things need to be retained. So if we mix with water, it will be helpful for the growth.
Yeah, and in naturally in our body, the cell stays in aqueous environment, right? So we want to mimic that. So that is why hydrogels are. Okay. Any other questions in understanding the logic behind using these three polymers as a bioink and how each of them have their own roles to play? Any queries on this other than what Murli asked? Okay, moving forward. So this scheme shows how the printing is done, and then how the steps in the printing are involved. So if you see, the composition of the ink is 10 weight percent gelatin, 0.5 weight percent alginate, and 2 weight percent fibrinogen. So which is among these, which component is highest in weight percent? Like in this bioink composition, which component is having the highest weight percent? Yes,
Gelatin
yes, is gelatin, right? Why do you can you think why they have kept gelatin as the highest one?
for mm, structural component to maintain the shape after printing.
Um, that's good. Okay, that's good. You're nearby, but I think they have done it so that they can print, because gelatin, as I told, is shear thinning in nature, so it will allow very nice formation of filaments. And once the printing is over, when you get the structure that you need, you can just wash off the gelatin. So that is why they have kept gelatin as the highest component, because other two components, alginate and fibrinogen, they do not have that shear thinning property. If they would have kept alginate as the highest one, then printing would not have been good. They would not have got good structures, good shaped structures out of it. Got it? So that thing also we need to keep in mind when you are making a bio ink, is to make an ink that is actually printable. We have to ensure that, means it should maintain its 3D structure, it should not collapse. The filaments are uniform, they are stacked well one over the other. Got it? And they have kept the temperature of the ink as 28 degrees Celsius. Again, why? Anyone? Can you think of the reason why they have kept at 28 degrees Celsius? So what was the phase transition temperature of gelatin? Yes, we just discussed. What was the phase transition temperature of the gelatin? It was 35 degrees Celsius, right? So any temperature below 35 degrees Celsius, gelatin will be in a gel state. So that is what they wanted. They wanted to make the like the ink in kind of a gel state, so that it becomes, it gets extruded out as something of a solid filaments. And then these filaments can stack well over one over the other. And since here we are only relying on temperature to give rigidity to the entire model, they have kept the bed onto which the printing is done at 4 degrees Celsius, so that the structure does not melt. Again, I'm choosing my words very carefully so that you understand the rationale behind using temperatures here. Why they have used such low temperatures. Got it? Any queries here? Why did they use a cool bed for printing? Why did they cool the bio ink to 28 degrees Celsius? Any questions here in understanding that logic? Yes.
I'm saying after preparing bioink, they will take it the bioink at 28 degrees Celsius. They bring it down to
Yes. Yes,
how they
yes,
will
yes.
bring, ma'am.
So they can use a refrigerator or ice or something like that. So it's not that difficult to do it. Even if the printer they developed, if they have some Peltier heating and cooling system, itself. Like the printhead can have the heating cooling systems, and you can accurately modulate the temperature at which you want it to be cooled or heated. So yeah, that's not the challenge. Like the printer can do it, or manually you can use ice or refrigerator to do it.
And what about the 4-degree Celsius support?
Here also you can a Peltier heating or cooling system can be used to cool the support. So yeah, support means the bed. So if you remember the first image of the 3D printer that I showed you, the support is actually this print bed. This print bed has, usually in the commercial ones, they have some um, this is called Peltier heating system or cooling system. It's a very efficient heating cooling system that is available till date, and there is no time lag per se for the cooling or heating to start. So this bed has that system connected to it, and you can manually enter the temperature that you want through a software. It will read and start the cooling. So this print bed is, in this study, is cooled to 4 degrees Celsius, and the print head, this is the print head into which the bio ink itself will be loaded, that can be cooled to 28 degrees Celsius. So that's the condition in the 3D printer that they have maintained for printing this bio ink. But my question is, did you understand the rationale behind choosing the temperatures at 28 degrees Celsius in the print head and 4 degrees Celsius in the print bed? Did you understand the rationale behind that?
They wanted
If
to
not,
have a solid substance printed.
yes, yes. Yeah, yes, in very simple words, you said it right. They wanted to make sure that the filaments that deposited did not deform, they did not melt. All right. And the main why they want to make sure that, because the main component of their ink is gelatin, and gelatin has a phase transition behavior. Below 35 degrees Celsius, it is gel, and they want to maintain that gel consistency. That is why the temperatures are kept very less than 35 degrees Celsius. Okay. And this is only physical cross-linking that is happening. Please mind, the entire structure is based on physical bonds. So if you increase the temperature, the entire structure will deform. All right. So whatever like spatial distribution of cells that you want, that will be lost. And we have to and that is what that is not required, right? So yeah. Now the temperatures are maintained, they have done the printing. So after printing, how do they make sure the bonds that are that is being made, that is now permanent? How did they make sure of that? So they did post printing processing. So how they did post-printing processing, they took the 3D structure out from the printer and incubated it for 30 minutes in calcium chloride solution. And this calcium chloride solution also contained a very small amount of thrombin. Why? Anyone? Come on, look at the other two components of the bioink and tell me, why did they use this incubation step?
To convert fibrinogen to something.
Hmm. Fibrinogen will convert to fibrin gel. And what about calcium chloride? How it will help?
Interacts with alginate, ma'am.
Yes, yes, that's correct. That's correct. So now this into this bath, there will be formation of chemical bonds or covalent bonds. Thrombin will is an enzyme. It will enzymatically react with fibrinogen to form fibrin gel. So it's a chemical reaction that happens whereas CaCl2 will release calcium ions. They will ionically cross-link alginate into alginate hydrogen. Okay, but ionic cross-linking is much stronger than the physical bonds that is made through temperature change. All right, but although calcium chloride is also doing a physical bonding, but it is relatively stronger. So these two components are actually going to help in making the structure more permanent. Now, what else is going to happen if you incubate this structure? What else is going to happen to the bioink? What will happen to gelatin? What will happen to gelatin if you incubate the 3D printed structure into this bath?
Can you repeat, ma'am? What?
What will happen? See, alginate will get cross-linked using calcium ions. Fibrinogen will convert to fibrin gel in presence of thrombin. But what will happen to gelatin in this bath?
I think it will get more. What is the temperature of that
Yes, that's the
solution,
good
ma'am?
question that you asked. That's correct. So that's a very good question that you asked. If the temperature is low, like if they used a cold solution, then they can retain the gelatin, but their objective is not to retain gelatin. All right. So they must have used the bath at a higher temperature, higher than 35 degrees Celsius. Then what will happen?
Ma'am, what is it?
They must have, their objective was to remove the gelatin, all right, eliminate gelatin. So they
High
must have,
temperature.
yes. So they used a bath that is at a higher temperature, and into this what bath, what happened? Gelatin got removed. All right. So now the after the post processing, you have the 3D structure made up of only two components, like theoretically speaking, alginate and fibrinogen. Although 30 minutes is a very long time, so I expect entire gelatin will get dissolved out. I expect that. If they have kept for a smaller period of time, then maybe some remnants of gelatin will be still there, but 30 minutes is a very long period of time. So that is what they did. You understood the 3D printing process and the post printing process. You understood that. So first they printed using three components of the bioink, gelatin, alginate, fibrinogen, and with it they mix cells as well. I forgot to mention, but it was understood. So these are the cells here. All right. This pink strands are the gelatin. Okay, and this is alginate. I'm just marking it so that you understand it clearly. And this is fibrinogen. All right. So they printed it. Consolidate step is actually the post processing step where they put it in incubation in a bath containing calcium chloride and thrombin. In that bath, what happened? Gelatin got dissolved. See, gelatin got eliminated. There is no more gelatin in this structure. All right. And now this structure is ready for maturation step. By maturation, I mean they will now put it in cell culture media and allow the cells to grow. So you see the morphology of the cells, they have now changed. They will start spreading, adhering to the substrate and spreading and then increase in number. And that is how they made the dermis layer. And then what they did after a few days of culturing, they um, seeded epidermal cells on top of the dermis layer. So these are epidermal cells, okay, and made dermis plus epidermis, that is full thickness skin model. All right. Any queries and understanding whatever was discussed in this slide? This was the main heart of the paper. Okay. If you understood this part, the rest is like smooth ride. Any queries? If you want me to repeat again, I'll definitely repeat it. There is not an
Ma'am.
issue. Yeah.
Mm, Can you name any analogs for these components, ma'am?
So, all right. So if you don't want to use gelatin, you can directly use collagen, because collagen also shows similar type of phase transition behavior. Okay, but collagen is slightly more expensive. Slightly means very, very expensive than gelatin. You can buy, say, 100 grams of gelatin for, say, 4,000 rupees. But if you buy collagen, even few mgs, like 10 mgs, maybe it will cost you 40,000 rupees. Okay. So collagen is more expensive, but it will do very similar. It will show very similar behavior like gelatin. Collagen is also another issue with collagen is that um, it will show shear thinning only when it's used at a very high concentration, like 10%. But since it's very expensive, people limit its concentration to very less one, like 1% or so. So if you use very less concentration, even gelatin won't show shear thinning behavior. So yeah, there is a disadvantage in using collagen, but if you see biologically, collagen is much more biocompatible than gelatin. So there is a trade-off. You have to decide based on what is possible for you. Then alginate, if you don't want to use alginate, then there is another polysaccharide that shows very similar type of cross-linking behavior that is called carrageen. Okay, kappa carrageen are there. Then there are different forms of carrageen. So that they are also derived from algae, and they can be cross-linked in presence of ions, divalent ions like calcium. Okay. Um, but again, they are not shear thinning in nature. Okay. Next, polymer that is their fibrinogen. So fibrinogen, I can't think of any substitute of fibrinogen right now, because this chemistry, that it will form fibrin in presence of thrombin enzyme is very unique. Okay, so if you want to just substitute fibrinogen based on the presence of RGD sites, that is the site that cells recognize and adhere, then you will have several choices. Okay, you can use elastin, like, and there are various other proteins too that will have these RGD sites. So you can choose any other protein, or you can modify a synthetic polymer to contain RGD site and then use it. Okay, so yeah, so these could be some potential alternatives or analogs for these components. But again, the concentrations you have to optimize at what concentration you want to use that analog. Then you also have to optimize the printability of that ink, whether or not that ink prints into a stable solid structure or not. So those things again plays important role in making sure that you successfully achieve your target. All right. Any other questions? Everyone understood the concepts discussed in this slide, because it's very important for the rest of the paper to be understood. So any questions?
No, ma'am.
No? All right. What about others? Vaishnavi, Harsha, Guru.
Ma'am,
Are you
actually, I
guys?
joined late. That's why I have to watch it again and then I'll ask if I'll have any query.
Okay, okay. But if you want, I can repeat like for the parts that you missed, it's totally fine with me. So that you understand the rest of the lectures. That's why. You can
Yes,
obviously
ma'am.
watch it again.
If you can give a quick review, then it will be great.
Yeah. Okay. 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 So glad you asked. So here I was discussing that the three components of the bioink, the three main components of the bioink is gelatin, alginate, and fibrinogen. And they are used at different weight percent concentrations. So gelatin is used at the highest concentration, that is 10 weight percent. Why? Because it is the component that will give the shear thinning nature to the overall bioink, and that will overall help in extrusion process. All right. And like it will also help in retaining the 3D structure, solid 3D structure after the print is over. So that is why they have also used a temperature of 28 degrees Celsius, which is much less than the phase transition temperature of gelatin, is 35 degrees Celsius. So the thing with gelatin is that it will form a gel if you keep it at a temperature 30, less than 35 degrees Celsius, and it will stay in solution form if the temperature is higher than 35 degrees Celsius. So they wanted to maintain the gel-like consistency of the entire bioink. So they kept it at 28 degrees Celsius, the entire bioink, and they print it on a substrate which itself is maintained at a much lower temperature, that is 4 degrees Celsius, in order to make sure as the printing process completes, it will take some time, right? Few minutes or maybe hours if you're printing very big structures, that during that time duration, the entire structure should maintain its solid like behavior. All right. So keeping such a low temperature was much more important, okay, when you're printing big structures. Otherwise, what will happen, the bottommost structures will start melting, and the new layers that are being deposited on the top of it will not find support to get printed on, and it will ultimately cause collapse. So that is why the temperature choice is made in this way. Now, note the entire structure that is now being made is physically bonded. So, uh, it is gelatin's structure that is being physically cross-linked right now, and entire like credit goes to gelatin at this stage for maintaining the rigidity of the structure. Alginate and fibrinogen, they are still in uncross-linked state, so they cannot contribute to maintaining the solid structure. So what they did a post print processing. How they did it? They made a solution which is composed of zero point 0.1 molar calcium chloride solution plus thrombin. So thrombin is an enzyme that will enzymatically react with fibrinogen to form hydrogel, that is called fibrin hydrogel, whereas calcium chloride will help release calcium ions in the solution, and these ions will now interact with alginate chains and form ionic crosslinks. Okay, so in this way, this bath is helping chemical formation of chemical bonds within the bioink. Also, it is incubated for 30 minutes. So it's a very long period of time for eliminating any remnants of gelatin that must be there. Okay, so they have washed off the gelatin, and the bath is not kept at a low temperature. It must be at a higher temperature. So maybe greater than 35 degrees Celsius, because you remember they have cells too. So in this bioink, they have cells too. So cells need 37 degrees Celsius at least to survive. So the bath now must be kept at a higher temperature to enable live cells, right? To make sure the cells don't die. So and so in this stage, after post processing, what you get, the bioink or the structure that you made, 3D printed structure that you made, only have the alginate and fibrinogen components in it. So If you see this figure, the four diagrams that are made, you see the first printing step is there, in which the four components cells, alginate, fibrinogen, and gelatin are mixed together. Then the second step is the post-processing step or consolidated step. Here gelatin is dissolved and only the alginate and fibrinogen are left behind, and they are cross-linked with the cells entrapped in the 3D matrix. And then what they did, then they did the maturation step. In maturation step, they had to use cell culture media to provide nutrients to the cells and necessary growth factors, so these cells thrive and proliferate and migrate and all in the 3D matrix. And then what they did, they added, so this is the dermis layer. So the dermis layer, they used cell type that is fibroblast cells. These are the resident cells of the dermis. And after dermis layer is formed, after a few days, they seeded epidermal cells on top of it. And in this way, dermis plus epidermis, a layered structure that is natively observed, that is formed. Okay, now this structure after secondary maturation is the complete skin structure. Okay, so quickly, I summarize this entire slide. You understood, Vaishnavi? If you have any
Yes, ma'am.
queries. All right, okay. So moving forward with the results that this paper has shown. So you see they have made very beautiful structures out of their bioink that they have made. And to showcase how good their bioink is, they have shown the pictures of these structures. The first structure is that of two wells. They have made two wells, and these wells are watertight. Means they have put some blue colored dyed blue colored water into this wells, and they show that there is no leakage of water from the wells. So the bioink is able to make, or the bioink plus the parameters, bioink and the parameters like temperature and all together, we call it 3D printing parameters, together are working well. Okay, so they are very well, so they are two parts of a very big puzzle, and you have to fit them together at the right position in order to get good results. So that is what they are showing, that they have fit it well, and they have been able to make 3D structures which are watertight in nature. Okay, then they have also shown that their ink is able to make very well complex structures. So they are showing that they have made honeycomb structure. Okay, so it's very complex, as you can see, and all the hexagonal patterns are very nicely observed, right? So hexagons has didn't become circles, it is a hexagon, as you can see. So that is what they are showing, that their bioink is able to make very good complex structures. Then they are also showing that their bioink can make overhangs. Means this is overhang, this part, there is no support here to lift the structure up. All right, and even the gravity is not dominant here to cause deformation. That is called overhang. So whenever people 3D prints this kind of overhangs, they try to put some support on it from the bottom so that it stays in place. But here they are showing that their bioink is able to form overhangs without any support. Okay, and the structure is efficient on its own. Okay, it's stable on its own. Then in this picture they are showing how the path of the ink, and how they are like very well like following each other. So this pink lines, they that is showing the path of the extrusion on the substrate. And I'm also showing that like in this picture they're also showing the filaments are quite smooth, all right, and continuous in nature. So there is no disruption in between the or within the filaments. All right. So these were the results. Any questions?
Ma'am, it's for the plan is to prepare a skin, right?
Yes, yes. Plan is to prepare a screen, but we have to first like see how well our bioink is, how good bioink you have
Okay.
formed, right? And then you can compel the audience that yes, my bioink can form an complex structure like a complex architecture like skin. So first you have to compel the audience that who are reading your paper that your bioink is doing a pretty good job.
So they
That is
gave
why,
us an example
yeah,
of
I'm talking of 3D structures.
cat-like structure.
Yeah,
Okay.
yeah, yeah. Okay. Now, I think my, can you guys see the next slide? I think my screen hanged for some reason. Can you guys see the next slide? Slide
No,
number
ma'am.
14? No, right? Wait, let me share my screen again. Can you guys see the slide number 14 now?
Yes.
Okay. So this was the previous slide, slide 13 that we just saw. This is your 14th slide. Now they have showcased that yes, our bioink is performing very well. Now what they have done, they have tried to show the compatibility, biocompatibility of their bioink. Means they are trying to show the effect of the bioink composition as well as the 3D bioprinting conditions that they have used, like the low temperatures and all, and the post processing. So all those I'm terming it as a bioprinting conditions, their effect on the cell viability. The cells were there, right, inside the bioink. So how that is affecting on the health of those cells. So this is the first image that they have taken on day 0 immediately after printing. So they have used in this experiment NIH3T3 fibroblast cells, and they are transfected with GFP. Okay, they have transfected with GFP. GFP, the full form of GFP is green fluorescent protein. So these cells can inherently release a fluorescent protein that is GFP. So it will give a green color fluorescence, okay, in presence of specific excitation light. So there is an exciting light and it will get green fluorescence protein. It will give green light that is in the range of 488 nanometer wavelength. So they have used these cells for this experiment. And what you see in day 0, the green color dots, they are the cells and they are round in structure. Okay, they are round in structure. Also, you see these cells are very well um, positioned within the 3D matrix, very well uniformly positioned, like in every corner of the image, you can see a cell. So the bioink that they have made have uniform distribution of the cell. The cell number that they have used is a good estimation as well. So that has made this possible, uniform distribution of cells. Okay. Now after 7 days, they did post processing on day 0, and then they kept it in a culture media, cell culture media. And after 7 days, when they imaged the same structure, they see that the cells have now elongated. Okay, they have elongated. So fibroblast has this kind of elongation type of morphology. In general, if you see image of a fibroblast cell, it you will see that. So similar kind of morphology is now observed. So they have like long protrusions. Membrane, plasma membrane have long thin protrusions, and they try to communicate with each other by getting in contact. So in this way, after 7 days, the cells have spread out, increased in number. Definitely it shows that the cell density has increased within the matrix. So what this shows as a conclusion, that their bioink and the 3D bioprinting conditions have a very good effect. Okay, very good effect on cell viability. It is not compromising the health of the cells. Cells are still alive and will be able to do its job, like secrete specific type of proteins, show specific morphology within the 3D matrix and all. Okay, so this experiment tells you that. Any questions? Got it? Got the experiments. These are very basic experiments. Okay, this is the paper 2017, and these experiments nowadays are routinely done by every lab all over the world. So if you ever join in tissue engineering lab in future, you have to do these experiments. Like either you or your seniors have done it already, and you will be next in line to learn these experiments. So you have to understand the rationale behind how these experiments are designed, and why the cells are chosen in that way, and what kind of microscopic technique that you use to capture these images, what kind of result would you expect after imaging. So these kind of ideas you would need later on if you ever decide to join a tissue engineering lab afterwards, after your current degrees. Okay, any questions? If not,
I have a question.
mhm.
Ma'am, at day zero, the cells are naive, or it is matlab, born that day, or how this protrusion of cell membrane or cell, uh, what I can say, is done within 7 days. Why not in day one or day zero?
So have you ever worked with cells before? Just a general, I want to know. Then I'll
No,
explain.
ma'am, not yet, not yet.
Okay. So, so very basic thing that they must have done previously. So let me add a page. Oh, no. All right. So what they have done is first they culture cells. So they are using fibroblast cells. So I'm explaining the general protocol they have followed. Okay. So they have cultured fibroblast cells, culturing in a monolayer. So they use a flask like this that looks like something like this with a cap, and it forms a monolayer of cells into this flask. Now, what they do, they remove these cells. How? They do a step called trypsinization. Okay, this in this step we use an enzyme called trypsin. Okay, and uh, these cells are now no longer in a monolayer. They are in a solution form, and the cells have lost their morphology, lost morphology. And whatever communication that was happening in between those cells, all those are lost. These cells are now form round kind of structures. This step also add stress to these cells. So you have to be very careful in trypsinization, means you don't have you cannot put trypsin for a very long period of time, and also the temperature and all that you use during this step is very crucial because the cells are stressed out here. At this step you can kill the cells as well. All right, you can decrease the overall viability of your cell. Then after trypsinization is done, you make the bioink. You add the cells to your bioink. Now, preparation of bioink can also add stress to your cells. How? So you use a hydrogel component, that is in this case gelatin, fibrinogen, and alginate. So I'm calling it as hydrogel for now for your understanding. And with this, you add the trypsinide cells, right? These two components are mixed together. And this hydrogel is usually viscous in nature. All right. So you have to use something like a syringe or a piston to make a homogeneous composition, right? To make a homogeneous mixture of these two things. So when you do that, when you mechanically mix cells with the hydrogel, you again add stress to the cells. So these two steps are already stressful enough. All right. And if you are not very careful, and if you are like doing excessive of trypsinization or excessive of mixing, you can kill the cells. And if you don't do enough of trypsinization or enough of mixing, again, you have to reach an optimum thing in both these cases. So that will come with experience. You will see after hundreds of failures, you will get that good thing. Okay. So once these two steps are done, then you print it. Now here printing also you are using temperatures that is much less than the optimum temperature of 37 degrees Celsius. So there you're using 28 degrees Celsius, and so not 280, sorry, pardon my handwriting. It's 28 degrees Celsius and 4 degrees Celsius. Again, these cold temperatures, what they can do, so they will slow down the metabolism of cells. These, so at cold temperatures, cells don't die. Only thing is that they stop functioning that well. Means they slow down, they become very lazy. Okay. So in this step also, the cells are not very healthy here. All right. Now you do the post-processing and then you incubate in the environment that is healthy enough. So what you do, you add cell culture media. So this has all the nutrients and the growth factors required. And you give proper temperature of 37 degrees Celsius and pH of 7.4. Now this is the healthy condition here, in which the these cells, the first step, in these first steps, the cells were cultured in these conditions. Okay. The cells that were cultured in first step and the last step, that the conditions that you are giving, they are the best conditions for cell growth that you are giving in the last step. Now you're asking why not the cells have like proliferated in day one, right? Like after day one, why did you have to wait for day 7? Now you think in this entire process how much the cells have undergone stress. All right. And in order to revive again, to come back to that healthy condition, it will take some time. They didn't give a time lapse image. If they would have given a time lapse image, you could have seen that over a period of time, you will start seeing that, let's say in day one, 10% of the population will start proliferating and coming to morphology. Then in maybe day two, rest 20%. All right. Then day three, 30%. In this way, it took almost day 7 for all the round morphology cells to convert into the spread out morphology, that is like the membranes and all, cell membranes and all, they protrude, right? They come out. So those, that morphology took time. Okay, that took time to up to 7 days. So it's a slow process. And if you must know, the cell cycle also plays an important role. So human cells, they do not proliferate as fast as bacterial cells. Like E. coli, it will proliferate in 12 hours or maybe less than that. But human cells, it takes almost more than 24 hours. And if the conditions are not good, it can take even 48 hours. So, yes, that is why it took time. Got it? Vaishnavi, got it?
Yes.
Mhm. This was a detailed discussion, but I hope it was worth it. Okay. Then the next discussion that they have given in the paper is to explain what impact did the printing process had on the cells. So here, when you're doing extrusion, as I already explained, the shear stresses are the main forces that is going to be detrimental for the cells, nothing else. The ink that you used is very biocompatible, that is not toxic, but the process itself can be toxic to the cells. So they wanted to show us th- that the printing parameters, etc., that they use, they are very favorable. So they did a very good calculations of shear stress. They calculated the value of shear stress, the minimum one and the maximum shear stress. They gave a range of shear stress range, and they showed that this range is very, very less shear stress value, and it cannot impact their cells health. So you know how to calculate shear stress. This is a very, very like common equation that you know from your like general physics classes. Shear stress, that is unit is Pascal, is equals to viscosity into shear rate. All right. So here the shear stress we are talking at of the shear stress that the cells will experience towards the walls of the nozzle. All right. So nozzle is like this conical one, and here these are the walls at which shear stress is maximum. So there is a gradient. Think there is a gradient. There is no linear thing. So the walls is the maximum, and at the center it's minimum, like that. So if I could like approximate the how the gradient would look like, it will look like some like this. At the walls, I'm not drawing a very good diagram here. I'm sorry, but I hope you understand what I'm trying to say. So here at the walls, the shear stress is maximum. At center, it's minimum, and again, it will be maximum, something like this. Okay. Then there's a viscosity that will play an important role, and the shear rate. What do you understand by shear rate? Shear rate is nothing but dV by dy. Just general knowledge. You can go back and search in Wikipedia. You will get the entire explanation of this formula. All right. So this is the formula that they use. So they did some rheological experiments. All right, to determine the value of shear rate. Okay, determine the value of shear rate. So how did they determine it? They determine it using this formula. Here, they knew the value of Q. Q is the flow rate. They have already set in the instrument. R is the top. So this is the cone, right? So R is this is the bottom part. This is the top part. R is the radius of the bottom part of the cone, and the top part of the cone. So that is the radius they already know of the nozzle, and the n they determined experimentally. So from this, they determined the value of what is shear rate. Determine the value of shear rate. Okay. And then what they did, they put the value here and determined the value of tau. Now, for viscosity, they didn't calculate the exact viscosities. Okay, so there's a, what do you say, constraint here. The material that they're using is not Newtonian. So Newtonian fluids like water, they have a fixed viscosity value. Whereas the material that they are using, or usually what I work with, are viscoelastic in nature. So the value of viscosity changes, okay, with conditions. So I'll not get into too much of details. So just for your knowledge, viscosity that they have kept as arbitrary, like um, eta T and eta B. All right, they have kept it as arbitrary. No, no, sorry. They have calculated viscosity values. I'm sorry. Pardon my previous explanation. They have calculated the values of viscosities. Okay. I think they did it again rheologically. So rheological experiments gave them the value of viscosities as well. And then they compiled this particular table for, so this is the minimum, this is the maximum. If you look at the geometry of a conical nozzle, you will see, you expect to see that minimum of viscosity will be, so minimum shear stress will be at the top of the nozzle, whereas as the nozzle becomes much more conical, here at this bottom stage, as the cells come in, the walls come closer, so the shear stress will be highest at the bottom, whereas shear stress will be minimum at the top. So you think of this table as a minimum and a maximum boundary that they're giving. So they have then calculated the shear rate. I told you how they calculated. So using this formula, they calculated the shear rates. So the maximum is around 52, around 52 per second, but whereas the minimum is 0.1 per second, then the maximum viscosity. So see, viscosity will change. It's a shear thinning material, all right? So that's why its viscosity is changing. When the shear stress was maximum at the bottom, the viscosity is quite less. It is 0.1 or almost. Whereas at the top, when the shear stress is minimum, at the top, the shear stress is minimum, then what happened? The viscosity is the maximum. Then they use these two values and into this formula, viscosity and shear rate, and calculate the values of respective values of shear stress. And what they see, that um, the stress values that they obtained are very less. Okay. From literature, from previous literature, what they found is, if the shear stress values are greater than 5,000 Pascal, then only it will have some impact on cell viability. Then only it will be detrimental enough to kill the cells. But since the shear stress values for them is like 12 Pascal and 51 Pascals, which is much less than 5,000 Pascal. So that is why they're explaining that their 3D printing conditions are not detrimental to cell health. Got it? Any questions here?
Ma'am, whether that 5000 Pascal is specific for this cell they are using or for living cells of human body?
So this is a
Pro.
general value they have taken from the literature, and they're assuming that it's going to be applied for all types of human cells,
Okay.
or maybe cells in general. Like you can use bacterial cells as well, but yes, you're correct. With the size of the cell and the makeup of the cell membrane and all, it might vary. Like, but you need a general approximation sometimes, to give you an idea, like how good is your thing, right? How good in comparison to others that others are doing. So they give a general value, but you can use this later on as well as a, what do you say, underline. If you're doing 3D printing or 3D bioprinting yourself and you want to test how good your conditions is on the cell health, you can use this benchmark. You can use this threshold that you cannot exceed 5,000 Pascals, because it is already reported in literature and people accept this value. So you can use that as a rationale to continue in your research. Okay.
Okay, ma'am.
Mhm. Now, till now, what we have discussed, there is no talk of skin yet, right? There is no talk of skin yet. Till now, in last two slides, we just saw the how good is their bio ink, did they have any did it have any effect on cell health or not. Okay, so there is no discussion of skin yet. Now the skin part will come. So just a quick recap for those of you who are not aware of how the skin looks like or what are the layers of the skin. So skin has two layers predominantly, the dermis layer and the epidermis layer. So hypodermis is not the layer of the skin, but rather it's a fatty tissue. Okay. And below hypodermis, there is the muscles. So if you go from top to bottom, that is the entire makeup of your skin that you see. So this fatty tissue basically helps in, accumulating excess energy and also insulation. It works. So these two are the layers of the skin that does all the functions of the skin. And epidermis is again divided into approximately 5 layers. All right. So stratum lucidum is not present, is only present on your palm and sole of your feet. Okay. That is why you see your palms and your sole of your feet is much thicker in nature in comparison to the rest of their body. So, but if you want to generalize the layers of your epidermis, so these are the 4 layers: bacille, spinosum, granulosum, and corneum. Right. So add stratum in the fronts. That is the name nomenclature. So these are the layers of the epidermis, and you see skin houses various kind of secondary apertures as well. Like there is hair follicles, then there is oil glands or sebaceous glands, right? Then there are nerves and blood vessels in the dermis layer. There is also some muscles and like various secondary structures are there. But in this study, they have only focused on mimicking or designing or 3D bioprinting these two layers of the skin, okay, with the primary cell types. So dermis has the primary cell type that is called fibroblast cells, okay, and epidermis has the primary cell type that is called keratinocytes. So these are the two primary cell types they're working with. If you want to make things more complicated, you can add more and more cell types. Okay. And like our body is so complicated, right? We haven't been able to, fully mimic it. So there is a lot of opportunity now. But yeah, it was 2017. They have limited technology back then. It is 2024, almost like how much? I guess 4 or 5 years has gone by. So yeah. Yeah. I guess 3 years, no? Sorry, I'm very bad at math. Don't mind me. You do the calculations and let me know. Okay, so this is the structure of the skin. Okay, very easy. Now they started with printing the first layer of the skin, the bottommost layer of the skin, that is the dermal layer. All right, and they are showing these two images. These two images are histological images that they called. Okay, and they have dyed it using an dye that is called Mason's trichome. So uniqueness of this dye is like you can see the nucleus of the cells and the ECM of the tissue colored in different colors, like distinctly, because they are stained different in different colors. So they are very uniquely observable. So they have done two samples. For sample one, there is no cells. All right. So they have just bioprinted the ink without cells. And in sample two, the bioprinting is done with fibroblast. And then they have showed the images at day 12 of culture. So day 0 was the printing, and from that day 12, like 12 days you add. That is your day 12 culture. And what they show is that in the second sample where there was fibroblast, the cells were able to, very nicely um, spread out and attain the fibroblast like morphology that I called here. Then also there is secretion of collagen. All right. So the collagen is shown in blue color thingies, and the dark blackish color structures, those are the cells because they has nucleus and all, a cytoplasmic component. And another interesting thing that is to observe is that various fibroblast cells and the collagen, they have formed a layer like this at the top. Okay, so that's a good thing. Why? Because after all, you're going to see the epidermal cells on the top. So when the epidermal cells are seeded, they will have a very good uh, contact with the fibroblast cells. Okay, and they will start communicating much faster. So it's a good thing. Other things that to note is, what do you say? Cells are very uniformly spread out. All right. So fibroblasts, the collagen that you see here is coming from the fibroblast cells. In our bioink, there was no collagen. There was gelatin, even there is no gelatin. There is only alginate and fibrinogen. The collagen component that you see here is secreted by the cells. So the cells are functioning well in this 3D environment to secrete this amount of protein. And yes, it took a very long period of time, but it did, but the cells are still functioning well, even after 12 days. So that's a good result, right? So yeah, so this is how they made the dermis. So it took them 12 days to make the dermis layer to achieve this kind of a morphology. Then what they are going to do next? They are going to seed the epidermal cells to make the epidermal layer. So epidermal cells, here is the keratinocytes. And they have compared the overall structure. So this is their 3D bioprinter skin. This is their two-layer 3D bioprinter skin. They have compared it with the structure of a human skin. So maybe a discarded sample of a human skin from a hospital or somewhere, and they stained it with the same Mason's trichome dye, okay, and compared the image of the stained human skin sample with that of 3D bioprinted skin. Okay, and what they observe is that, like the human skin, there is a dermis layer. Like the human skin, there is a junction that is called dermis epidermis junction or dermal epidermal junction. All right, so there's a junction that uniquely separates the two layers from one another. And also there is a epidermis layer at the top. Okay, so that is the three layers. So the two layers with the junction that is fully mimicked, morphologically mimicked, all right, in their bioprinting approach. And it looks, and this structure is a representative of what the human skin looks like. Okay, you see, the epidermis is very well stacked over one another. This is called terminally differentiated epidermis. You see the thickness of the epidermis that they have achieved. It's very similar to that of the human skin. All right. So this is around like uh, obviously long. I don't remember the number of days that it took for them, but I think it's like 24 days, I guess. I guess. Let's check the paper once, okay? So after 24 days, I'm assuming they have got, or maybe over a month of culturing, they have got this kind of 3D bioprinted skin. Okay, makes sense? Now, this image or this result only shows that there is a morphological similarity. What about functional similarities? Is there a functional similarities or not? Like, is there secretion of those specific proteins which are known to give the functional properties of the skin? Are those proteins are getting expressed or not? How do you confirm that? So they did another experiment that is called immunostaining. So here they use antibodies. They can be monoclonal antibodies or polyclonal antibodies. These antibodies are produced with specific binding capacity to a specific type of protein. And on the Y end, so antibody, looks like this, right? So on the tail of the antibody, there is some fluorescent dye tagged. And this fluorescent dye will actually help us locate the binding regions. That is the like very simple, very rudimentary explanation of immunostaining. Okay, so these are the sites to which antigens will bind, and this will be the dye. Here, our antigen is the specific proteins that we are looking for, that supposed to get expressed only when it is required. Like there is some proteins which are known to be involved in giving the barrier function to the skin. So they are trying to check whether those proteins are expressed or not. Then there are specific proteins of the dermis layer that gives the structural rigidity to that dermal layer. So those proteins, whether they are expressed in their bioprinted skin or not. So what they observe, let's discuss those. So this panel is for the dermis layer. This panel is for epidermis layer. Okay. So you see, this is the human skin. This is their bioprinted skin. Again, they have used the human skin as the control. All right. So you see in the bioprinted skin, the proteins like collagen 1, collagen 5, fibrin, laminin 332, laminin 7, all of these are expressed. Here, note the green regions. The blue regions are usually the nucleus of the cell. So they use a dye called DAPI. So that stains the nucleus blue. So note the green regions. Those are the locations of the proteins. And you see in all the images, wherever there is green in the human skin, there is a green region in the bioprinted skin. Have a look at it. So this shows what? This shows that all the proteins that are natively expressed in the dermis layer of the skin is also getting expressed in the bioprinted version. All right. So that functionally gives us proof, that gives us proof that the bioprinted skin is functional or has a functional dermis layer. All right. Now, all these proteins play an important role in your human skin, right? So they are showing that. They are making some noteworthy observations now. This laminin 332 are only found near completely new the dermis epidermal zone. Okay, the junction that you see. So you see, this is the junction. This is the line that separates the epidermis. This is the epidermis. This is the dermis. This is the line that separates. So when this protein is getting expressed, it gets expressed only near this junction. Similarly, here also it starts to getting expressed. This shows what? That this junction is getting newly formed in the bioprinted structure. That's the proof of that, that this junction is getting formed in the bioprinted structure. All right. Then I already told you the proteins of the dermis layer, the collagen 1, 5, fibrillin, elastin, collagen 7, these are all getting expressed in the dermis. So that's also proof that the dermis is now functional. Look at the epidermis zone. Okay. So in the epidermis zone, they have now used antibodies with dyes that fluoresces red. So look at the red regions here in this panel, in the epidermis panel, look at the red regions. So what you see, so they have marked the zone with a dotted line to make the image much more clear for the readers. So, and above this white dotted line is your epidermis. Okay, below it is your dermis. Okay, and the blue dots are the cells, position of the cells. All right. So cells can be of any origin. So don't confuse the blue dots. Notice the here the red lines, here also the red, but for flagellant, notice the green regions. So depending on what type of dye that is conjugated with your antibody, you will get specific type of coloring in your sample. All right. So here cytokeratin is a protein that is observed to be expressed in the epidermis layer. All right. observed in the epidermis layer. So you see in the human skin, most of the region is red, and, and in the bioprinted skin also, this is quite good. This is prominent. All right. Red regions are there. That means cytokeratin 10 is getting expressed in the epidermis. Then phlegarin here also is a component of the epidermis. It is getting expressed. You see the green line in the human skin, also in the bioprinter skin. Like very similar positions, they are getting expressed. That's a very good sign, right? Then there is a protein called lauresin. So that gets expressed near the stratum corneum. All right, the topmost layer of the epidermis. Stratum corneum is the layer that you can see right now on your body. That is the stratum corneum. And the cells that are there are mostly dead. So they are dead actually. And every 28 days, you shed this layer of skin, and the bottom layer then it comes up and forms new cells. Okay, and replenish the stratum corneum. So the formation of stratum corneum is a mark that epidermis is fully formed. So expression of this protein that is associated with this layer, stratum corneum, shows that this layer has successfully formed. You see the slightly red color lines, okay, in the bioprinters panel as well as the human skin panel. All right. So through this experiment, all right, they have shown that all the proteins associated to the function of the human skin are getting expressed. So this is not a complete list. This is only a small list of all the proteins that can get expressed. There is a large number of proteins that are there in our human skin, and but obviously definitely due to time and the resources constraint, they could only show a few of them, but enough of them to help the help convince the readers that functionally they have made the bioprinter skin which is functional. Okay, is functionally viable. All right, got it. Moving on. So they also did
One
a
doubt, ma'am.
T. Yes, go ahead.
Ma'am, in epidermis, you mentioned multiple layers, right? 5 layers. We are using skin stem cells to make those 5 layers. How we
um
generate those 5 types, different types.
so if you look at the structure of the skin, the bottommost layer, stratum basilae, it people have shown that there are stem cells here, okay, in this layer. And but they are very less in number. Stem cells are very less in number, and they get only activated when needed. So they help replenish the lost number of cells, but they're only present in the stratum basilae, right? So you have to give them specific conditions for these stem cells to actually get activated and start proliferating, right?
Okay, they would have done this, right? That's how they
So they
generated.
no, so you're asking how did they make this 4 layers, 5 layers in their model, right? That is your question.
Yes, ma'am.
Without stem cells. So they are not using stem cells, by the way, right? So how did it? That's a very good question. I forgot to mention the like protocol. So let me. So your dermis is ready. Let's say in 12 days. Okay. Then on top of the dermis, you seed keratinocytes. These are the main cells of epidermis. All right. You so this is your structure, two-layer structure, and there is, let's say, a well like this. You submerge this two-layer structure into cell culture media. Now this stage is called submerged culture period. and you do it for few days. Okay, you can check the paper to check how many days, for how many days they did submerged culture. So here again there is keratinocytes and there is fibroblast at the bottom. After few days of submerged culture, let's say I have done, like, I also work with skin, so I have done in my experiments like for 14 days almost. Okay, but you check the paper for how many days they did submerged culture. So after submerged culture, you did what you do to get that terminal differentiated structure. What you do air liquid interface culture. How? So you just reduce the volume of the medium in such a way that the topmost layer is exposed to air, sorry, and the bottom layer, that is your fibroblast layer, is submerged fully. Only the topmost layer is exposed to air. This is called air liquid interface culture. And you do it again for a few days, like maybe um 7 days or something like that. I have done this for 7 days. And what this condition does, it kills the cells. All right. Because there is no media, right? Whatever media that it is getting, whatever media that the epidermal layer can get is at the bottom. The cells that are at the top, they basically stop. They just die, right? And they just make structures like this. I've done some microscope images and I've seen very smooth structures like this. That was quite interesting, right? So that is what we want. We want a dead layer of skin. And if you do TEM, so they have done TEM imaging, transmission electron microscopy imaging, you can actually see features, specific features. Okay, these are very, very tiny, like the nanoscale features, all right? Like nanometer scale features that are signature to each of the layers of epidermis, all right? And that will confirm that 5 layers or 4 layers of epidermis are formed. But the key step in doing in making the 4 layer epidermis or 5 layer epidermis is this step, air liquid interface culturing. If you don't do this, then you won't get that and like layers of epidermis, right? That is how they did it without using stem cells. Got it?
It is one way of developing, ma'am, or other methods are there?
This is the only way that I saw, and it is very well established one. I didn't see yet any other method that people are using to get this kind of a structure for skin. It
Okay,
is a
ma'am.
very, very old method and very well exploited for making skin tissue.
Okay, ma'am.
So there is some TM images in the paper. All right. I didn't show it in the slide today because this is very complex to understand. But if you want, you can go and have a look. Okay. And while you read the paper, but for the sake of keeping this lecture simple, I didn't include it. So was that, like, what they did is, like, in the images they just tried to show very tiny features of each of the layers of the epidermis and said that yes, they have made epidermis. But as a researcher who is working in this area, I felt I didn't understand those images very nicely, to be very honest. Okay, I have to myself go back and maybe look up some anatomy books with histological images and all to actually confirm, like, that is how the epidermis looks like. Okay, so I'm not very sure about how to explain those images to you. That is why I left it for the part, so I'm being completely honest here. Okay, but for your curiosity, you have the paper with you, go ahead, download the paper and have a look later on after the lecture is over. And the last thing that they did is like show the readers that their ink or their bio ink is capable of printing very large structures, like in the centimeter range. Okay, the centimeter range, it is able to print very large structures. Although this thing is not related to your functional characterization or morphological characterization of skin, I think they just did it for like showcasing their work, like to show how good their work is. That's all. So they printed a human ear. As you can see, this is the CAD file, and after it is printed and cultured in cell culture media. So this is your cell culture media. It usually looks red like this. Okay. And since it's a hydrogel, the entire makeup of the bioink, maximum makeup of the bioink is hydrogel. So it absorbed the cell culture media and turned pink in color. So you can see the human ear that they printed is like pinkish. So that's all. That is the paper. And that is how they demonstrated the power of this technique, extrusion-based 3D bioprinting technique, to make a human skin model, right? They developed their own ink, they developed their own machine, okay? And then they printed two layers of containing two different cell types, showed the readers through good characterization techniques that the skin is successfully formed. And yeah, there you go. That's the end of the paper. So any questions if you have, let me know. It's a 7-year-old paper. It's a very, very old paper. Hello, can you guys hear me?
Yes, ma'am.
Yeah. So any questions so far? Actually, I'm done with the explanation, so the stage is open for questions. Did you guys enjoy uh like hearing to the story that they have made? Did you like the concept? Did you find it interesting?
Yes, ma'am. But what is the motto of their work, ma'am?
Motu?
Of their work, they printed, right?
Yeah, that's it. Yeah.
That's it.
Yeah, but you now feel that's it. When they did it 7 years back, it was an impactful work. That is why it came out in a very good journal. Now people are doing much more complicated stuff. Like they are not using just single cell types, they are mixing different cell types. Also, they are not using only one type of technique. They are what do you say, mixing with other techniques, right? They are um, combining multiple techniques with each other. So now things are becoming much more established, but still we are not actually there yet to completely mimic our body's tissues. All right. Some parts we can mimic so far, but everything together, like add the nerves, add the blood vessels, add the lymphatic system, add secondary structures and all. So we are not there yet, right? A lot of research is underway and people are doing it. And yeah, hopefully, maybe in 10 more years, I don't know, maybe more than that, because our body is very complex. God has done a brilliant job, and we as biologists, we can only try to mimic it. We cannot actually make a human being yet. Okay. So there, I remember there was a talk back in when I was doing my undergrad. So there was a professor who came from IGIB Institute in Delhi. It's a very good CSR institute. So she was working on genomics. So I don't recall her name, but she's a very well established principal scientist there. So she was telling that she has combined genomics with Ayurveda. So I asked her, like, can we actually mimic the entire body yet? Like, can we make a human being yet? All right. So she told me, like a car, we have only yet started understanding the principle of engine, how the engine works. Okay. Or how the window shield works. We haven't been able to assemble all the parts yet. So that's a very good explanation. And I didn't have any knowledge of research or tissue engineering at that point of time. I just randomly asked her because I found her research very interesting, and I just had that question like, is it possible yet? So a lot of research. So I have completed my undergrad in 2020. So it's before than that. So only 4 years of experience I have in research till now. So I myself have a lot to learn. And you guys are just starting out. You're just learning the field, learning about this field. So when you will start out, you will see the entire scenario getting changed.
Okay.
Ma'am, are there such labs near Lucknow in which we can do such type of experiment?
Definitely. There are, like, I'm working with a collaborator in Lucknow, in PGI Lucknow. They do tissue engineering work. Like, they work with stem cells and they work with iPSCs and all. Like, there's so many labs in Lucknow. You can try out to get some internship or something like that in PGI or in CSIR labs are there. Like, CDRI is there. Then there's Toxicology Research Center is there. That is also CSIR lab. So I know of these three institutes. These are government institutes, and there are professors there. You can approach them, you can mail them, and try to work with them if possible. So yeah, there are people. So tissue engineering is very mainstream now. Everyone is working some part of tissue engineering in their work. They are incorporating it in their part. If you see the basics of tissue engineering, it comes from cell biology. All right. And cell biology is the very, very basic course that you all study in biotechnology, right? So whoever knows cell biology, they somehow try to learn tissue engineering as well and incorporate it in their work.
Okay, ma'am.
So go through the paper. All right. I have just tried to explain you guys the work that they have done in a very rudimentary fashion. More details are there in the paper. You can go through the materials and methods section to understand the experiments, how they did. Okay, the exact number of days they cultured, the exact number of cells, and also you can have a look at it. But when you read the paper, I hope you'll understand it better now. Okay, that's the point. That's the point of taking up a paper, so that you understand the technique, why they used it, how they made the ink and all. That's my job. If you guys understand that, I'll say that I have done a good job. Okay. And I'll soon post an assignment too. It will be similar, like MCQ types, like the last one. So it will hardly take like maybe 10 minutes if you understand the subject. So to complete it, so complete your assignment, submit it. I'll see you guys next week. So next week we'll start understanding another type of 3D bioprinting technique. All right. So, okay. I'll see you guys next week then. Right?
Okay, ma'am. Bye, ma'am.
All right.
Thank
Bye.
you, ma'am.
Mm-hmm.